Hello students and welcome back to the chapter, and in this module we shall be having a look at transposons. Now let us study what transposons are. Transposons are commonly called as jumping genes. Now why they are called as jumping genes? We shall have a look at it in a brief time. So transposons are sequences of DNA that can move or transpose themselves to new positions within the genome of a single cell. So transposons have the capability of changing their places in the DNA or the genome, and these changes can create certain differences in the phenotype or the genotype of the organism. So now let us take a look at the history of transposons. The first transposons were discovered in sea maize or maize, which is commonly called as maize or corn, which was done by a lady known as Barbara McClintock, and she discovered the transposons in sea maize in the year 1948. She also discovered that 50% of the genome of maize consists of transposons, and she was awarded the Nobel Prize in 1983. So the first transposons were discovered by Barbara McClintock in sea maize or corn, for which she was awarded the Nobel Prize in the year 1983. Now let us take a look at this cell. Here I have a bacterial cell which is showing me the bacterial chromosomal DNA. It will also show me another extra chromosomal DNA which is known as plasmid DNA. And I have a segment of chromosomal DNA which can jump into the plasmid DNA. Now this is a transposon. Here the gene that has jumped from the chromosomal DNA to the plasmid DNA is the gene that is conferring antibiotic resistance on the bacteria. So this is a type of transposon. Now let us have a look at the transposons that are found in humans and primates. The transposons found in humans and primates are allosequences, which are approximately 300 bases long, interspersed and repetitive DNA molecules. So these are the transposons that are found in humans and primates. Let us move ahead and take a look at what transposons can do. Transposons can create phenotypically significant mutations and can alter the cell's genome size. Now this depends on the type of the transposon. So let us take a look at the types of transposons. Here the first type that I have is retrotransposon. So here we have the DNA or the genome, and which is showing me a segment of DNA as a retrotransposon. So here this retrotransposon undergoes transcription to form RNA. Now this RNA will be reverse transcribed into a single stranded DNA by the enzyme reverse transcriptase, and this single stranded DNA will be converted to a double stranded form later, and this double stranded DNA will now be inserted into new position in the genome. So here we have a new copy of retrotransposon which has been inserted into a new position in the genome. So this is how the retrotransposons work. Now the insertion of retrotransposons take place via the formation of RNA intermediate, and the method is the copy and paste method. Let us take a look at the next type of transposon. Here these are known as DNA transposons. Now DNA transposons require no RNA intermediate. Here we have the target DNA, and this target DNA will be cut by the transposase enzyme. This enzyme will now insert the transposon at the cut place or the nicked place, and the open nicks or the breaks in the DNA will be later sealed by ligase. So this is the method by which DNA transposons change their positions in the genome. And what do they do? How do they do that? They do it by the cut and paste method. So retrotransposons work by the copy and paste method, whereas the DNA transposons work by the cut and paste method. So in this module, we have studied transposons, and in the next module, we shall be having a look at plasmids. So till then, take care, study well, and bye-bye.